So hello, everyone. We will thank you very much for joining today's webinar. We will just wait for another half to 4 minutes to allow more people to join. Just a second. Thank you. Okay, so I suggest that we are going to start. So my name is Anna Jantke-Busse. I'm product manager at Invitek Diagnostics, and I am very happy to introduce our guest for today, Sarah Handem. So Sarah will today provide us with her exclusive insights in the field of food testing. So some words about our speaker. So Sarah did a Master of Biochemistry at the University of Coimbra in Portugal. And afterwards, she did a PhD in molecular biosciences at ITQB Nova in Portugal as well. So Sarah has now a life science research experience for more than a decade. So she has a lot of experience in various fields in molecular biology, genomics, microbiology, and biochemistry. So, and having a track record also in techniques such as DNA, RNA extraction, and real-time PCR, next generation sequencing. Sarah is today focusing on the field of food safety. And since the end of last year, she is employed at Jerónimo Martins, where she is responsible for the food testing services of their clients. And this is also what she is going to introducing today. So Jerónimo Martins is a company based in Portugal that operates in food distribution and specialized in retail sectors. So more about this, Sarah will tell us about shortly today. Yeah, so just some technical things. So after this talk, as usual in our webinars, we will have a Q&A session. And I kindly ask you to type your questions to the Q&A module that you see at the bottom of the Zoom surface. So since the chat function will be deactivated during this talk. So yeah, thank you very much again. And Sarah, so now I will hand over to you. We are looking forward to your talk.
Okay, thank you, Anna, for your kind introduction. I hope you can hear me well. So, good afternoon, everybody. Today I will talk a bit about GMO testing and what we do here at the Jerónimo Martins DNA Lab. I will start by introducing a bit the laboratory and what do we do in general terms, and then I will be focused specifically on the GMO testing. So, let me just... So we are, just 1 minute, okay. So we are a molecular biology lab and we perform food analysis, mainly based on DNA, so DNA analysis. And we are a lab that was one of the first among food retail companies. We opened in April 2021 and we are accredited by the ISO regulations. We are located in, cannot see. So we are located in Portugal, more specifically in Azambuja, which is near Lisbon in the Ribatejo zone. And we are located inside the logistic platform of the Jerónimo Martins company, one of the logistic platforms. So our team is small but very good and beautiful. And we have Carlos Santos, which is our director, the Quality and Private Brands Develop Global Director. And then we have Sofia Nogueira, which is our lab manager, so our lab head. And then we have three lab technicians, me included, and Claudia and Rita. And then we have a lab assistant, which is Miguel, and also Andrea Fernandes, which is our technical assistant and performs the administrative management work. So our customers nowadays are several stores. So we have Pingo Doce and Recheio stores in Portugal. We also have the Biedronka stores in Poland and also the ARA stores in Colombia. So these are the private brands of the group Jerónimo Martins, and we test the products from all of these companies. Since early this year, we also provide our services to external clients. So we not only provide services for the products in the company, but also to other clients that want to test their products. So why does a food company decides to create a molecular biology lab? So this is not very common, and there were many reasons for this. And one of those reasons was to increase the authenticity control capability in order to reduce the probability of fraud in the food. Another aim was to create upstream pressure in the food chain, and by doing that, we ensure that the products have high quality ingredients. And also, in cases where we need to react quickly to some urgencies that arise, we can make sure that we can do the analysis quickly and ensure a higher support in decision making in the company and other clients in general. So what we do is that we analyze any feed or fruit product that contains DNA. And we focus mainly on more processed products that usually have higher economic value. And why DNA? So we choose DNA because DNA is present in all organisms and at the same time is unique for each organism. So we can reach to the species or to the subspecies level. And it is also highly resistant, so it is not indestructible, of course, but is highly resistant to several mechanic and other types of processes. So if we want to reduce food fraud, one of the things we need to know first is what is food fraud. And food fraud is defined as the addition or substitution of one or more species in a product with the intention of deceiving the consumer, usually for economic gain. And maybe some of you can remember some cases of fraud. For instance, in 2013, we had the cases of the substitution of beef meat by horse meat, and also the substitution of codfish by other less valuable economically fishes. So when there is food fraud, there are several risks that are associated with that. And one of the risks is that we lose the traceability of the production chain. Also, we can have introduction of antibiotics or pesticides or other chemicals in human food, possible poisonous species, and also species that are not poisonous by themselves, but they can produce toxic compounds. So these are some of the risks that are associated with fraud. So how do we do these analyses? We focus on two main techniques. One is next generation sequencing, and this technique reads all the DNA sequences in the sample. So with this technique, what we want to do is to determine the species that are present in a sample. And when I say species, I'm talking about ingredients. So that have DNA, of course. So if we have meat products, we can detect if it's there swine, bovine, chicken, turkey, or any other type of meat. Regarding fish, we have cod, pilchard, salmon, tuna, so every fish that you can think about. For plant, the same rational, crustacean, and mollusk species also. And soon we want to introduce the method to detect insects that are becoming more and more a case of interest in the food production chain. The other method that we use is the qPCR or real-time PCR. And this method, what it does is to search for a specific DNA sequence in a sample. So it doesn't read all, it reads a specific sequence that we want. And with this method, what we want to do is to assess the presence or absence of genetically modified organisms in a sample. And these nowadays are done for plant products, plant-based products. So we have a working procedure to treat all the samples, And this procedure is common at a certain point to both the NGS and the GMO screening, but at some point they diverge. So I will show you briefly what are the workflow that we do. So we start by getting the samples, the food samples, and these samples need to be logged in, and we need to take photos, introduce information about the product, like the type, the batch, the expiration date, so the client, so all these information that are available about the product. And this is very important because we need to have traceability, and we have to make a chain of custody to know where our sample come from. After this sample registration, the sample enters the lab 1, and in here we do the sample evaluation and processing. And in here we evaluate each sample and determine what is the best homogenization protocol for the product. And these protocols are different among, so there is not a defined standard protocol for the species. It depends if you receive a ham that is in slices, or if it's a big chunk of ham, for instance, if we receive a tea that is just if it's just dust, or if it has small pieces of fruits or other plants. So every sample has its own way of being homogenized and processed. After this step, we don't have the food anymore. We have a sample, and this what we do with this sample is to extract the DNA that is present there, and to do that we do the lysis of the cells that are present in the sample. And this extraction or DNA extraction can be done by robots, so it's an automatic process, or it can be done manually, depending on how many samples we have. After extracting the DNA and getting rid of everything that we don't want, we quantify the DNA samples and dilute them to a desired concentration. After this, we do the PCRs, and in here, both the NGS and the GMO screening have a PCR step. The difference is that the NGS pipeline has a conventional PCR and the GMO screen has a real-time PCR. So we have this room, the lab 3, where we prepare the reagents mix. So in this room is DNA free. So it's only the mix that is prepared inside this room. And once it is done, it enters another lab, that is the lab 4. And in this lab 4, we do the addition of the DNA to the mix and run the PCRs. So as you can see here in this image, we have a person there doing the mix, and then in here we have another person that is adding the DNA to the mixture. And we finally put the tubes in the thermocycles and rerun the PCRs. After this, we can do electrophoresis, and this step is only used for the NGS pipeline. So when we need to do NGS, we need to run the PCRs in agarose gels. And in here, we assess the DNA bands, if we have the bands, if we don't have, what are their intensity, so on, so on. If we continue with the NGS, the next step is to perform the DNA sequencing. This is in lab 5. And in here, we construct the libraries and we load the chips. So we have these micro chips that have millions and millions of sequences. And then the sequences are sequenced and we have results. And then we have a step of analyzing all those results. In the case of the qPCR, so we run the PCRs in lab 4, and then we have to do the analysis. And in here we have softwares to analyze the result curves, and we need to check if we have amplification curves and compare them with control. So do the data analysis that I will talk more in the forthcoming slides. After all this, we go to the office where we do all the report analysis, and we issue the statements of conformity and do all the lab planning and lab management and so on. So this is like a straightforward circuit. We start in one room and we never go back. We always go forward to prevent contamination and other things to happen. So I told you that now I will be talking more about the GMOs, and to do that, I think it's important to introduce the theme a bit. So we know that in nature we have genetic variations occurring in all the organisms. We are not static; we are always evolving and changing. And these naturally occurring genetic variations throughout the years have been chosen for breeding. So when we want to generate food and feed, we select specimens with desirable characteristics such as resistance to disease, tolerance to herbicides, and others. And we do this breeding, and then we generate food and feed, right? So this is the naturally occurring process. But nowadays we have gene technology, and with gene technology we can edit artificially the genomes, so modify them in order to achieve those desirable characteristics. And when we do that, we have genetically modified content that we can produce food and feed with. And it's important to notice that in the European Union nowadays, only GM crop plants are approved. So it's the only type of specimen that it's possible to modify. So there is European Union legislation to control and to assess GMOs, and GMOs are defined like ipsis verbis, like organisms in which the genetic material has been altered in a way that does not naturally occur by mating or natural recombination. So it's a different modification that what will happen in naturally over the time course of life. And genetically modified food or feed are defined as food or feed that contain or consist of such genetic modified organisms or are produced from it. So the European Union has a very strict regulation about GMOs, and their approach focus mainly on four topics. So we have safety, we have labeling, traceability, and freedom of choice. So any GMO to be approved to be placed in the market needs to be authorized, of course, and there is a very thorough risk assessment of the product in question, of the genetic modification in question, to assess if it's safe before entering the market. If it's approved, it enters the market, and after being on the market, it is continuously monitored to be continually assessed. So it's not a thing that happens before and then stops. It is continually being monitored. Once it is in the market, there are a few rules, and the main important ones is that all food and feed with more than 0.9% of genetic modified content must be clearly labeled in the product, and this is very important. And also, all food and feed with content must be traceable to its origin. So, with all these things, the European Union wants to address the four pillars of their approach. And of course, once everything is done, the risk assessments, the labeling and everything, the consumers and all the professionals have freedom of choice to choose what they want. And this is the most important thing. So currently, we have more than 100 GMOs that are authorized for food and feed uses in the European Union, and these are divided by these categories. So maize, cotton, soybean, sugar beet, oilseed rape, and also at a much lower level, potato. I think there is only one event that is approved for potato, but it's starting to be out there. So mainly, why is so important to test the GMO in plant-based food and feed products? So mainly for three things: to continue to monitor that there is no cross contamination between GMO crops and non-GMO crops. To ensure that we have product compliance, so what is in the product is equal to what is stated in the labels, and to provide the consumers and the professionals with an informed choice. So they have the information at their disposal and they can choose what they want to consume and to buy. So just to give you an idea, the top 5 GMO crop productions countries are the USA, Canada, Brazil, Argentina, and India. I think they get more than 90% of the GMO crop production worldwide. And as you can see, I put this picture just to show you that in Europe, the only 2 countries that produce GMO, that has cultivation of GMO crops, are Portugal and Spain. Portugal at a much lower level than Spain, but we are the only 2 European countries that at this point produces GMO crops, and only maize. So the routine testing of GMO, it's a reality in our lab. We do it every day, and for that we use the InviScreen GMO detection kit by Invitek Diagnostics. And briefly, this kit is a qPCR method for the qualitative screening of GMOs in food and feed plant-based products. It has the most, or it detects the most frequently used transgenic regulatory elements in genetically modified plants. It uses fluorescence channels, FAM, VIC, and X. It has a plant-specific primer set to control the reactions, and we have overall 0.1% GMO content detection, so limit of detection, which is a very good limit. This kit targets 3 main targets. We have the 35S promoter from the cauliflower mosaic virus. That is one of the targets. We have the nopalin synthase terminator from a bacteria present in soil, that is Agrobacterium tumefaciens. And we have the 35S promoter from the figworth mosaic virus. And these 3 targets are present, so they constitute most of the genetic elements that are present in genetically modified plants. So the tests that we do, we target these 3 targets. So once we have an amplification curve that is positive for one of these targets, and after comparing with controls, we can assess if the products are positive or not for these elements. In the case of the first target that I talked about, the P35S promoter from the cauliflower mosaic virus, we also use an additional kit because when we have positive results for this target, we cannot automatically identify the plants as GMOs. And why is that? Because the cauliflower mosaic virus, it's a virus that naturally infects plants. So when we detect this target, we cannot be sure whether we are looking at true GMO positives or false GMO positives, because they are indeed infected with the cauliflower mosaic virus. So we do this additional test where we target a region of the virus itself, of the virus genome, and after getting the results, then we can conclude something. So in case that we have a positive result for the P35S promoter and a negative result for the CAMV virus, we can attest that that product has the genetic modification. When we have a positive results for both targets, we cannot ensure whether or not the genetic modification is present. And this is important because, and I told you before that the GMOs are modifications that does not occur naturally in nature. And in here, it's one of these cases. So when we have the GMO, we only have this piece that is the promoter that is inserted in the genome. And when we have the plant infected with the virus, it's the whole virus. No, so it's not just a piece. It's the whole virus. So we need to assess the differences between these 2 in order to give a result with confidence. Also, for all the targets that we use, that we test in the lab, we always compare them with the GMO concentration standards between 0.1%, that is the limit of detection, and 1% because of the European guidelines. And after that, we issue the statements of conformity of our products. So we use these kits because it is suitable for the analysis of food feed samples. It is very friendly, user-friendly to use. It's fast because it's only like 2 steps. You just mix 2 things and then distribute. We have a multiplex reaction for 2 of the 3 targets. So it's 3 targets, but 2 of them are in the same reaction. We have the premixed reagents, so this means usually few protocol steps and also less probability of contaminations and everything going wrong. And also the method is very reliable, sensible, efficient, robust, specific, and reproducible. And this is not only what is stated by the brand, of course, but when we started using this kit, we did a verification study to demonstrate the performance of this kit, and we were very happy with the results, and we decided to use it. So what type of products do we test for the GMO screening? So I told you that it's plant-based products. And in here, we have several categories like non-alcoholic drinks, sauces and syrups. We have meat products, fishery products, because when we think of meat and fish, we can wrongly think that there's only meat and fish, but we also have plants in these type of products. We have sugar and sugary products, spices and condiments, pasta, pet food, products, fruits and vegetables, pickled vegetables, vegan products. We have pre-prepared meals, high-protein products, oils and derivatives, corn-based products, because corn or maize, it's one of the main crops, GMO crops that is produced worldwide. We have rice and all types of cereals, muesli, granola, and so on and so on. So these are the general categories that we test. These 4 categories, the pet food, fruits and vegetables, and vegan products, and also pickled vegetables, are the ones that usually have the higher incidence of GMOs because the main component it's plants. So the probability of finding in these types of products are higher than the others. So in here, I will just show you a brief example of analysis that we would perform. So imagine we have this product 1 that has plant-based ingredients, and after we have our DNA, our DNA from the sample, we do the qPCR, and together with the sample, we test 2 positive controls and 1 negative control. And this is the first step that we need to evaluate after a run. So we need to see if our internal amplification control is okay. And this is a control that is present in all the wells of the mix. So it's present in the mix. So it needs to be in all wells, including the negative control, right? Because the negative control has mix but doesn't have DNA. So we need to have an amplification curve. So in this example, I have 4 wells and I have to have 4 curves for this amplification control. And then we have the control for our DNA plant. So we need to make sure that we have DNA, plant DNA there. And in this case, we want to have 3 curves because the negative control doesn't have plant DNA. So it's okay. We have our 3 samples with plant DNA, so we are good to go to the next step. So one of the targets that I told you that we test is the Figworth mosaic virus, a promoter of this virus. And in this example that you can see here, we have the curved amplification curves for the controls, and our sample is negative, right there with the negative control. So we can conclude that our sample is negative for this GMO element. When we test for the nopalin synthase of the Agrobacterium tumefaciens, it's the same. So we have our positive control 1%, the 0.1%, and again, our sample is negative for this target in here together with the negative control. So we can conclude that our sample is negative for these targets. When we test for the P35S promoter, it's a different story. So in this sample, we have our positive controls and we have our sample amplifying in the middle. So it's a positive sample for this target, and it's between the 2 limits that we usually test. However, like I told you, we cannot be sure if we have a true P35F F35S positive, so we need to do the additional test. So when we go to test the CAMV virus, we see that our sample is negative for the virus. We have the positive control and the sample is here negative. So we can confirm that in this case, this product one is a GMO positive product, and in this case for the P35F element. So we have 4 targets or 3 targets and sometimes 4, and we can have multiple combinations of the results, right? So in here I have 2 other examples, but there are multiple combinations. So if I have a sample that is negative for these 2 targets and positive for these 2, I cannot, so my result is inconclusive. I cannot attest that the product has or has not a GMO event because it has both. It was positive for the original virus, and for instance, in this example, I have the product that is negative for all the targets. So this product is GMO negative for these events. So, of course, if we continue to make combinations like we did in preschool, we would end up with multiple combinations and possible results for this test. So after analyzing all the results, what we do is to issue the statements of conformity for the products. And these statements of conformity are always taking into account the client specifications. So we can have a compliant product, a not compliant product, or an inconclusive product for this test. And for instance, I can tell you that in the Jerónimo Martins group, the policy is to encourage the use of GMO-free ingredients in their private brand products. So this is a group policy, and other clients may encourage the opposite. So it always depends on the client. So as a final highlight, what I want to tell you is 2 main things. The first one is that our DNA lab here from the Jerónimo Martins analyzes any food or feed product with DNA. And like I said, we are not restricted to the products of the group. We can analyze products from any client at once. We do 2 main analyses, one to determine the ingredients of the products and another to determine the presence or absence of genetic modifications. And all the results that we report to the client always takes into consideration the product's ingredients list or the client specification. So this is not straightforward analysis. It always depends on what the client has in policy. And we aim in the future also to develop new targets for GMO screening to get a broad range of targets. Regarding the future of GMOs, I think the future is a bit uncertain at this point. If you start reading literature about this topic, you will find different opinions and texts about the subject. And I think that nowadays we have a lot of factors that affect the food. We have extreme climate events that completely damage food production. We have political conflicts, trade conflicts, population demands versus public perception, because it's also a very important topic. We have crop diseases emerging all the time that we need to cope with. We have the globalization, because like I show you in the map, for instance, the European Union does not produce a lot of GMOs crops, but it imports a lot. We have the globalization factor. We also have new biotech methods to engineer new GMOs. And we also have the factor of human genetics, which is very important. I can show you an example of these. So in the US, there is already an approved GMO event for swine meat. So it is possible to do it in animals in the US. And what the element that is approved it's the alteration of detectable sugars, a specific type of sugars in the surface of the cells. And this is important for people that has a specific syndrome that they make allergy to when they eat meat. So, and this disease is very common in the US, so they approved this modification in the pigs, so they can feed this type of people. So this is just one example. So I think that the important take-home message here is that in the end, what is important is to have transparent policies to enable consumers and all the professionals and everybody basically to make an informed choice regarding food or feed. I think this is the main thing that needs to be settled. So this is what I wanted to show you today. I hope you enjoy it, and I'm open for questions, of course. Thank you so much.